All right, so ending the last chapter, our last section of the last chapter, or this chapter, man, I can't get anything right to start this off. We're going to talk about Mendel and how Mendel didn't know what chromosomes were, but then Bavaria and Sutton came along and said, "Hey, we're noticing these chromosomes are going to be inherited," and this is about 50 years later. And so, what actually Bavaria and Sutton did was they talked about chromosomes being inherited, and they studied fruit flies for years. Fruit flies are a great uh, organism to study in genetics because they reproduce very quickly. They have very phenotypical characteristics, and so the study was continued by somebody called Thomas Hunt Morgan, who we're going to talk about in the next section. But just talking about the wild versus mutant trait, wild trait, that is what we typically see. So the wild, don't think like out in the wild, just think, hey, this is what we typically see. And the mutant, that's the non-typical trait. Ask me why they don't say typical and non-typical. Scientists have to be difficult. I don't know. So what they did was they were crossing again. This took a while. They took years to cross these guys. You can see they took a red-eyed female and a white-eyed male, and they crossed them, and they saw that the resulting offspring were all red eyes. So we have two different phenotypes of eyes here, and the resulting offspring were all red eyes, which kind of makes sense because again, we had the two different types of we should say phenotypes with the pea plants, and then that recessive phenotype is we should say masked in that F1 generation. The F2 generation, they again made these again for a little bit of incest. That's okay, they're flies. The resulting was again two out of the four were red-eye females, one was a red-eye male, and then we got this one white-eyed male, which is really cool. What was actually going on there was the eye color gene is on the X chromosome. You can see here in the F1 generation, you're going to get all red eyes because these females are going to give these red eyes to all of the offspring. When you cross them again, though, with a female that's heterozygous, you're going to get this one time we're going to get this male with a white eye. Now, I know you're probably going to like, Mr. Fradoff, what's the importance here? Like, what does this matter? This showed that chromosomes are going to contain genes or something, I should say. They didn't know about genes yet that are going to regulate the phenotype of the offspring. That was huge news. And I know you're probably again sitting there like, wow, chromosomes have genes that regulate who we are and what who we look like. Like that's crazy, Mr. Fradoff. Like who would have thought? I know that's what you're thinking, but this is really big. It's in the early 1900s, more than 120, around 120 years ago, 100, 115, whatever. That's huge news because we didn't know any of this. Just think about like back in the like early 1800s, people had no clue about what dinosaurs were. Like if you would have told somebody back then, like, oh, there are dinosaurs out there, like, oh, you're crazy. That's all made up. Like now it's like, okay, there's dinosaurs, duh. Like people didn't know about genes back in the 1900s. It's absolutely crazy to them that this is like oh my gosh, this is really groundbreaking research. All right, so what that leads to is we're going to talk about the dihybrid crosses, and we're going to talk about the law of independent assortment here. We touched upon this last chapter, but we're going to come back to it again. Law of independent assortment says that each factor segregates independently from each other. So you can see here, these factors are going to independently separate from each other. They're going to separate during meiosis, and they're going to not be inherited specifically together. Now, when you do a dihybrid cross, remember when we do the gametes here, you can see A and B. So these two aren't going to be inherited together. You're either going to get one of these A's or one of these B's. So the first combination is going to be big A, big B. Our second combination is going to be big A, little b. Our third combination is going to be little a, big B. Our fourth combination is going to be little a, little b. So we could have any one of these four genotypes here because crossing over can occur between these two. So you can get the big A, big B, big A, little b, little a, big B, little b, little a, little b. So I usually do this on the board. We'll probably do this together in class, but you can fill this out. Or again, you're gonna have your packet. You can just work on. Remember, this is because of meiosis and the independent assortment where it can go mom's chromosome here, dad's chromosome, or mom and mom, or it could be even dad and dad on this side. And you can get any one of these combinations of chromosomes. Oh, we're gonna stop. So I'm not going to go too in depth with this probability stuff because it's pretty easy if you can just do basic kind of math in your head. But I will go over like one or two examples. And there's a whole page of this in your packet. So Mendel's law of probability, mathematically measure of likelihood of genetics. Uh, whenever you see or whenever you see the word and, you're going to multiply. Whenever you see the word or, you're going to add, and that's going to come in handy later. But let me explain what this is going to be. So you can see here, it's like the sperm and the egg are like flipping a coin. So if you have like a heterozygous here, half the times if you flip a coin, you should get heads. Half the time you should get tails. Same thing with the egg. You should get heads half the time, or you should get tails half the time if it's heterozygous. So you can imagine here, if we flip two coins and we want both heads, it's going to occur about 1/4 the time. If you want a heads and a tails, you're going to flip the coin, and about 2/4, or you can see here 1/4 here and 1/4 here, or 1/2 the time you're going to get the heads and the tails. And about 1/4, you're going to get the tails and the tails. So you can see here that the heads and heads is 1/4, heads and tails is 2/4, and the tails and tails is 1/4. What does that mean, Mr. Friedhoff? Well, basically, what you can do is in your head is do these monohybrid Punnett squares very quickly, and then you can just like multiply them. So again, if you have a big B, little b, and you're looking for the offspring to be homozygous recessive, if you know that the chance of giving the little b is 1/2, and the chance of giving this little b is 1/2, you just multiply those together, and you're going to find this 1/4 probability. You can see that 1/4 is down here. And a cross between this, a big A, big B, little b, big C, and big A, little a, big B, big B, big B, big B, and I'm sorry, big C, big C. What's the probability of this? This looks like a really difficult question, right? Like that's a big hybrid or trihybrid Punnett square. But if we look here, just look at each individual genotype. So what's the chance of getting big A? I'm sorry, what's the chance of getting big A, little a between these A's? Well, that's going to be 1/2. If you do that monohybrid Punnett square in your head, it's going to be 1/2. What about the B's? Well, it's also going to be 1/2 if you just do the B. So again, in your head, do the monohybrid Punnett square for the B's. So we multiply that. Then the C's, well, it's also going to be 1/2. So what's the chance of getting this offspring from these two? If we just multiply everything together, it's 1/8. So you don't have to make this trihybrid Punnett square. You can do this math very simply in your head by doing monohybrid Punnett squares. Let's do one more. What's the chance of getting big A, little a, I'm sorry, big A, big B, little c from these two? Again, 1/4 here, if you do the Punnett square for the A's, do it in your head. Big B from these two, 1/2. And then little c, 1/4. So if you multiply all these together, it's 1 in 32. All right, so this is a way you can do a trihybrid Punnett square, or even a dihybrid Punnett square, and do it a lot quicker than actually writing it out.